I am Dr. Philip Macmillan, and part of my work is to look at science and to ask hard questions about science and to listen to people who are studying and have expertise in science. One of those is Professor Dolores Cahill, and some of the things that she says can definitely be controversial. As such, my recording on YouTube has been shortened, and you will be able to see a part of that interview, certainly where she highlights her background and her expertise. I think is important for everyone to know. If you wish to see the full video, the 1 hour and 22 minutes, and I certainly let her speak and share her thoughts on science as well as the law, you must then click on the link below. But to hear Dolores, the first part of our interview, please listen here. So, yes, Dolores, I think the first thing that I would like you to do is to do a proper introduction of yourself. So, I want you to tell us about your background and I want you to share with us your journey in terms of scientific research.
Thank you for having me on. I guess, Philip, my initial journey, we'll say, started when I was a child in Tipperary, and we lived on a beautiful farm and everything was perfect. And then a pharmaceutical company, Merck Sharp and Dome, when I was maybe 7, 8 years old, was not far from our farm in the valley of Slieve-na-Mon in County Tipperary. And then in the next spring, all of our calves, 17 of them, died, and some were born with 2 heads and 2 front quarters. And then for all of my childhood, basically, the case of one farmer and his family, John Hanrahan and his mother, was them taking on this scientific company because it was obvious that there was something that they were doing that was killing and poisoning the land and the animals, and he and his family and many families were sick. Now, during that process, my father was one of many farmers that supported him, and my grandmother and grandfather were really well versed in the law, including the real natural law, Brehon law, the ancient law. But I saw how the legal system just took money and there was no justice. So I was interested and visited Merck Sharp and Dome every year, even as a teenager, 14, 15, 16 years. And when I decided what I would do, I decided really the law anyone can learn. In a way, you just have to study. But to really understand the lack of scientific integrity, because it was obvious that the Merck Sharp and Dome, it was known that they could prevent this poison that ended up being a dioxin. They could have just put adaptions to the chimneys and that this wouldn't have poisoned the land. And they had the same issue in Puerto Rico 10 years before. And the Puerto Rican farmers took them like a decade to get them to stop the poisoning. And so it was gross negligence and willful blindness. So when I was 21, the farmer, John Hanrahan, won a Supreme Court case. So when I went into science in Trinity College, I did it in order to restore integrity in science. And I was taking science 1 year at a time. So that's a kind of an important element of my backstory. But also when we were, so also when I was 15, 2 employees of Merck Sharp and Dome came to our family and said that me and another family member, because essentially a lot of us, we weren't officially bankrupt, but of course people had no income because Avonmore and the milk collecting companies, they wouldn't admit there was a problem in the valley, but they stopped collecting milk in order to bankrupt the farmers. And then my father was in the IFA, the Irish Farmers Association, and they were helping the farmers against the banks, which I learned as a child how that process worked. And in 1977, my father was election agent for someone trying to get Ireland out of the EU, because the farmers and fishermen in Ireland knew that us joining the EU was a sellout of farming and fishing. It was destroying those industries. And TJ Maher actually won. And I was like 7 years old, 8 years old, putting up posters, but listening to conversations about sovereignty about the rights of indigenous peoples and about how big world organizations like the EU were trying to undermine sovereignty and the indigenous rights of people and access to their land and resources, which of course was entirely true. So then in 1982, two people came from Merck Sharp and Dome because they were saying only one farmer, John Hanrahan, had the issue. But my father wrote into them and described how, and I saw the letter, our animals had died as well. Now, what Merck Sharp and Dome did was actually put his letter in the canteen so that the people working in Merck Sharp and Dome would then create division because it would be public, right? And then after that, two people who worked in Merck Sharp and Dome came to my family and said that if my father stopped supporting the case and speaking out, that they would fund myself and my brother if we went to University College Cork, right? Mm-hmm. And of course, we immediately, even though we had very little money, and I ended up as 15 traveling in Europe and working in a factory in order to work for the summers while I was 15, 16, 17, and 18 to get the money to go to university. And we didn't obviously agree, and I went to Trinity. But of course, straight away, you know, my father looked at me and my other family member and said, you know, and obviously we said, no, why are you trying to bribe us? Why are you not trying to sort out the poisons that are making people sick and you know, making the animals sick? So then, then when I went to Trinity, I was in a lab, and this was about 3 years later, and there was two technicians that had spent the afternoon destroying evidence. And then I stayed in the lab for like 3 hours and took notes. And then at the end I said, you know, because they had said it was the Merck Sharp and Dome case. And out of our own money, we had paid for sample collection and vets to do the testing on the animals that were dead or sick in order to provide evidence for the case. And then I reported that to my dad, and that was reported in the case and one of the reasons for taking it over. So then when I went on to do the PhD, I went to study an area called brain tumors, but I was using tools called antibodies. And there were antibodies from a number of different manufacturers against the same protein. So it could be like PSA or CA125 or, you know, HE4, a test. But what people generally do, and this is important for the general discussion, Philip, is that people generally use one antibody for most of their career. And so the results are consistent, right? And then if you look up those papers and the other scientific groups around the world, the results look the same. But because I was coming to science from a very fundamental point of view, I ordered all the antibodies and I checked the publications from the different suppliers for the various pathology results of the slides. And it turns out that the antibodies for the same thing were given completely different results on the histopathology and tissue slides, right? Mm-hmm. So that made no sense. So I was asking people, you know, and pathologists and histopathologists, the people who do the diagnosis, how can it be that if you have an antibody to something that it gives completely different results and that nobody is checking? And they were more or less saying to me, oh, that's not an area that you want to look into. And the reason why I was obviously very interested in the law, and I said to them, this is important because I was dealing with glioblastoma and astrocytoma, so like almost fatal brain tumors. And if someone was diagnosed with that and they didn't have it, they were actually, you know, going to have significant treatment and surgery, right? So then you come into the area of the law because people are wondering, how do I know the law? So that if you're going to use an antibody and do research on something that could potentially diagnose somebody with a tumor where they would then go into an operation that could risk their lives, you have to make sure that you're doing all that lawfully. And the area of law that it is around people's inalienable or inherent right to life. And all science and medicine and research is based on, it's called the natural law. So it's high up, it's in the hierarchy of law. Yeah. I explain it, it's like a tower building with 12 stories.
Yes.
And the natural law is, we'll say, on level 11 of this imaginary tower. And above that is the area of conscience, right? Which is almost like there is a resonance of justice in the world. Mm-hmm. And the way I explain that is, if you have a baby that's 4 months old and can't speak, and they see an injustice, the baby will intuitively know that there's something wrong, right? And this conscience is also the area that's tapped into when you have a lawful trial by jury, that people listen and weigh the evidence, and it's based on, is something right or wrong? Is it moral or immoral? Is it ethical or unethical? But that whole area of law sits in the natural law. So the natural law is very simple. It's really 2 sentences. It's called act in honor, do no harm, and that men and women, people, have an inalienable or inherent right to life, travel, speech, privacy, and property. And those rights are with us every second that we're alive, and they're not given to us, they come from us, and they can never be taken away.
I'm going to pause you there a minute, Dolores. I mean, it's fascinating to hear that background. And that's what I really wanted to understand, where you were coming from personally that makes you take this kind of stance that is challenging the system. Because it seems to really be coming out of your own personal experience, your own personal journey that has brought you here. So, within that context, how did that play out when you were working in some of the top institutions like Max Planck and so on in Germany? Because you got some very significant positions there. How did that kind of positioning for integrity play out in that situation?
So no, that's perfect. So I guess just to go back then to the right to life. So I decided then, so I finished my PhD. I did it from November 1989 and I finished it in 1983. And then I got a job as I was writing it up in Munich for 2 years. And then I went on to work in the Max Planck and be a group leader there. But based on that experience in my PhD, I decided that I would then develop a technology that would check if the tools for diagnosis were accurate or not. And that, so I had spent like maybe 4 years in my PhD looking at the specificity and cross-reactivity of these antibodies. And then in Munich, I looked at the specificity and cross-reactivity of 2 more antibodies. So that was like a total of 6 years, right? Looking at just 3 antibodies. So then I'm an optimist, but also a realist. And I thought, well, I'm the only one doing this in the world, and I'm not going to live long enough, and be able to do it. And I certainly won't get funding for it. So I then looked at the research. The Human Genome Project was started, and one of the leaders in the world was Professor Hans Lehrer, who was Austrian and who was working in Imperial College in London, and he went to become the director of the Max Planck Institute in Berlin. And he had invented these high-content DNA arrays in order to map the DNA part for the Human Genome Project. And that was actually before PCR was invented. So we all know that the inventor, of PCR, he's well known now in the world, right? The Professor
Mullins.
Professor Mullins, exactly. I actually know the man who actually rang him to tell him he had the Nobel Prize. He was on the Nobel Prize committee. So PCR, so just to remind people, there was no mobile phones, there was no internet, right? The PCR was just invented. And what I wanted to do was to look at proteins and antibodies in large scale. Just at the time when people actually, and I sequenced using radioactivity and gels by hand, right? The old-fashioned method. So the technology that Hans Lehrer was developing for DNA was just emerging. And so when I was a postdoc in Munich, I then wrote to Hans Lehrer and asked, could I modify his technology for DNA and proteins? And he gave me a 1-year contract in the Max Planck. And in the process of that, there was a group that had, I was literally working night and day. In order to, I only had a few months really of a contract there. And then one of the group leaders wasn't able to work. And Hans came to me and said, will you find the funding for 5 other people and be their group leader? Now, I didn't really want to do that. I just wanted to do my work. But he said, look, pretty much you have to. There's nobody else. So I wrote 5 big grants for 2 or 3 million And in that year, I was involved in patenting the technology, because even though people might think patenting is wrong, if you publish something and it is useful, someone else can patent it and then block me from using it, right? So, and I donated back all of the that I got to the Max Planck and to my companies in order to progress the technology. So I invented then these high content protein arrays. And what you could do, we ended up with 250,000 clones on this array, and then we found 37,000 clones, expressed proteins, and then we ended up with about 11,000 different proteins. So, and that technology development took about 7 people, 3 years, and it was in the Protein Technologies group, and I was leader of the Protein Technologies group and the main person who did the writing for the patents. And this would have been in the years 95, 96, and then I filed the patents with the other co-authors in 96, 97, and then I set up a company, Protogen. And the reason why I set it up is there was such huge demand for people with antibody binding to use the technology that I couldn't just be giving it to my PhD students and technicians. That wouldn't have been ethical. So I set up a company, and we basically funded the company ourselves from making the high content protein arrays and people using them. So what these technology then was is that, say, Philip has generated an antibody by injecting maybe a cancerous tissue and done the initial screening on Western immunoblotting and mass spectrometry that was available at the time, but you didn't know whether it just bound the protein that you were interested in. So say maybe it's a prostate-specific antigen or CA125 that diagnoses ovarian cancer. So with our technology, if those proteins were on the array, one of 11,000, you could see if it was specific just for one protein, if it was, and then also that it wouldn't cross-react or bind to any one of the other 11,000. So that took me a number of years to do. And then when I started, you know, I was doing keynote, you know, I won many awards, and all 5 grants that I wrote all were funded. And I was nominated and applied for a BioFuture Prize that was only given to 8 people in Germany in that time. And, you know, about 350 scientists were selected to apply. And I was one of the 8 that got this BioFuture Prize. And part of that money was to set up my company. So I was given keynotes around the world. So I would have been in my mid-20s. And the technology was saving, you know, years and years of work. Right. But what I did, and as part, so, and also I got an award from the German government. So they actually asked me, would I at a high level advise the German government? Because I had so many grants that I was at the meeting saying, you know, there's a lot of the whole administration process could be made more efficient, you know, that you could just ask people what are the outputs of the research, such as PhDs or papers or jobs or prototypes or whatever. So they often took my advice. So that's when I very early got into advising governments around either innovation or technology transfer or benefiting patients, because what I was saying is there's too much money given to research that doesn't benefit patients. And also I then use the technology to look at autoimmune diseases. And people who have autoimmune diseases, their immune system is stimulated often to react to their own body. So we could just take a drop of blood, put it on the array, and you could diagnose what was the antibody profile associated with this person. So we ended up between my group in the Max Planck and Protogen looking at the blood of 20,000 people. And so I also did it very fundamentally, because if I was to study, we'll say, a disease of multiple sclerosis or alopecia, or if it was sudden adult death, right, that a lot of the time I found that the diagnosis was very sloppy, right? So that just, for example, with say in the multiple sclerosis group, I was saying to the clinicians how do you know that these people you're saying have multiple sclerosis? Because on average, then it took 12 years for someone to be diagnosed with multiple sclerosis.
Mm-hmm.
So the symptoms of autoimmune disease can often be chronic fatigue and other very generic symptoms. And it often happened that if they went to their local physician and the physician, had an interest in a particular autoimmune disease, or if they had symptoms of tiredness and they went to someone that was a general practitioner in Germany or a cardiologist, they would send them, with autoimmune related cardiology disease when it may not be. So what I did then was I spent about a year finding the key autoimmune groups in the world who were the absolute experts at defining the criteria for that disease. And then with them, we monitored people with that disease over, 10 years, taking blood samples at different time points. Mm-hmm. And when it was obvious that they were full-blown, autoimmune dilated cardiomyopathy or alopecia or multiple sclerosis, we would then study the late-stage samples, but also the early-stage samples before they had full-blown symptoms on these high-content protein arrays to get a profile of what people are at late stage, and what's more important is at early stage, because it turns out with autoimmune diseases, and in my company we did clinical trials with 20,000, is that if they are diagnosed early with the symptoms of arthritis or alopecia or autoimmune disease or whatever it is, if you look at their balance of nutrition and minerals and vitamin D and vitamin C, you can actually reduce the severity of the symptoms or eliminate them, so that we would say, and I've been lecturing, is that they may be predisposed to have a very severe autoimmune disease. But if they actually get the balance of vitamin D and vitamin C correct, they won't have the symptom.
Yeah. Actually, I'll pause you there. That's an interesting point, Dolores, because I remember for myself personally, I used to have quite severe eczema in high school going into university. And I don't know why I was looking at omega-3s. And I found that at that time I was taking extra supplements of omega-3s and the eczema completely disappeared. And then years later, I thought to myself, did I just imagine it? So I stopped taking it for, say, 6 months, and the symptoms came back. So it without a shadow of a doubt, each individual potentially has a risk. And it's almost as if we have to work out that risk personally to try and identify where it is. So for someone else, it's not that. It may be magnesium or vitamin D or vitamin C, but each person then has to be able to work that out. I'm assuming your work can't demonstrate that.
Oh, no, we were involved in doing that with 20,000 patients, right? So that was groundbreaking at the time, right? Because we were working with key, clinicians in the area. But exactly as you said, what I would say, I would not be a fan of like just taking multivitamins or whatever, right? So this is now, this is for 30 years. So the Nobel Prizes for vitamin D and vitamin C were 70 years ago. So really, when I was educated, which is, we'll say, almost 2 generations ago now, right? We're talking 40 years ago or so, right? Or at least as well that the people who taught me when I was in Trinity and doing my PhD, they would have been in university, 30 years before, right?
Yeah.
So, and the Nobel Prizes for the vitamins were at the time when they would have been in university, we'll say in the 1950s and 1960s, right? Mm-hmm. And I was in university, we'll say in the 80s and do my PhD in the 90s. So, of course, they were lecturing us about the importance of nutrition and things like zinc and, magnesium and food, right? And that the food had to be nutritious, which it was then, right? And, of course, that was very well known that you could actually reduce the symptoms, right? So which is what I did. But the thing was what my technology with high-content protein arrays, it was showing that, we'll say for multiple sclerosis, it took 12 years at that time for people to be accurately diagnosed. So I also worked with a company in America that were focusing on collecting blood samples already when I contacted them for 15 years. Mm-hmm. Right? And so that we were able to show, so the whole issue with this thing is if you treat them with supplements for multiple sclerosis and the symptoms stop, everybody would be saying, well, how do you know, right? And maybe the people that have full-blown symptoms are in wheelchairs have a different type of multiple sclerosis, right? Mm-hmm. So what we showed is that the people who had full-blown multiple sclerosis, we had the samples from 10 years before, that when they took the proper nutrition, their symptoms were alleviated. And we also showed that people who were early stage, because once we had our testing for alopecia or sudden adult death, autoimmune related, that then that test, the sudden adult death test, was immediately transferred in Germany because it was so life-saving. Yeah. That people could then be diagnosed as autoimmune related, and then Professor Felix had a process to dampen down the immune system, and they fully recovered, and they did not die, and they did not need heart transplants, right? Mm-hmm. So that was basically by using other people who had collected samples over 15 years, and by dealing, that was so I'm very strategic, right? By dealing with the top labs in the world, because our technology was so groundbreaking, that when you collaborate with the experts in the world, there is no question then that the patients that we were using were properly diagnosed, because a lot of the time they have a group of patients called a cohort, that those patients and families have a relationship with the research group and they consent.
Yeah. Can I pause you here, Dolores? Because one of the important things that is coming out, and I can see some of the comments, is that there has been such a negative association with the scientific industry, and the question about financial gain and issues about whether or not everything is only for financial gain, and whether or not there is a genuine interest in patients. How does the scientific community overcome that? Because at the moment, the public are now looking at the whole of the scientific and medical community in the same way. How is that fixed?
So I guess, Philip, that's a very good question. And the thing is, when I did my initial, so I was in the Max Planck maybe 95, 96, 97, and Professor Hans Lehrer and that whole, Max Planck were world leaders in the Human Genome Project. And so with many others, we decided that in order to fix this exact problem that's happening today, it's relatively easily fixed. And I was one of the people who helped write that grant, because when I was checking that the antibodies weren't working and my technology was able to check whether an antibody was accurate or not, I immediately said that if this grant was funded, that I would put everything that I had done into what's called a public center, and that anyone could order my tools of the high content protein arrays that cost a lot to make, but they were made available to people free. Order whatever antibody that I did on my publication and repeat the experiment. And if I was doing anything wrong or fraudulent, they could publish it and challenge me, right? So the grant became the resource center of the German Human Genome Project, and the German government funded 40 technicians over 10 years and made everything that was done in the Human Genome Project in those years we had to, and we're glad to transfer it to this resource center. And all of the tools were given freely all around the world to advance the technology, because we were being funded by the men and women who were paying taxes in Germany, right, which is the right thing to do. But that is how you then can check if a scientist is accurate or not, or a physician or a doctor, right? And the reason that you do it is there's often competing labs, right, and competing surgeons and competing doctors. So if someone, Philip, is in an area that you're doing research and you have planned and you've got grant funding to do an area of research that you've planned for 5 years, but if Dolores in a lab in Ireland or Berlin does something that makes your entire research program with your 2 PhD students obsolete, this way allows that if Philip says, I'm not sure if this is unbelievable work, that if you're required to put everything that I have published into a public repository, then Philip can go and repeat it. And if it's not accurate, he can then submit a publication and say, I don't think what she did. So why I now have a lot of standing around, why I came out in 2020 was saying that we need to get the things that were the main tools of the unlawful lockdown were the PCR testing and also this program that came out of Imperial College